Assalamualaikum and good day everyone. Today I'm going to talk about sequence alignment. Sequence alignment, or the comparison of protein and DNA sequence, is an important analytical method of applied bioinformatics for annotation of new nucleotide and protein sequences, construction of model structure for proteins, design and analysis of expression studies, and variety of other bioinformatics and biological experiments. Novel functionalities do not appear because of new genes have suddenly arisen, but are developed and modified during evolution. Given this situation, therefore, one may transfer functional information from one protein to another if both have a certain degree of similarity. However, this process must carry out critically because similar proteins may yet perform different functions. The similarity of two proteins can arise based on evolution from a common ancestor or independently of each other based on different ancestor proteins. Before analyzing whether sequences are possibly related, it is first necessary to define some terms. The term homology. Homology refers to the state of sharing a common evolutionary origin. We say two sequences are homologous if they have a common ancestor. There are two types of homology. First, orthology. Orthology refers to the state of being homologous sequence that arose from common ancestor gene during speciation. Second, paralogy. Paralogy refers to the state of being homologous sequences that arose from common ancestor gene from gene duplication. The definition of term ortholog and paralog in combination with function of protein and however subject of controversy. So in general, genome biologists call homologous protein from different species that have the same function, for example, corresponding kinases in signal transduction pathway in human and mice, as orthologs. In contrast, homologous proteins that have a different function in the same species, for example, two kinases in different signal transduction pathway of humans, are called paralogs. Homology is not quantifiable. Either two sequences are homologous or they are not. The identity or similarity of two sequences is, however, quantifiable. Identity. Identity is the ratio of the number of identical amino acid or nucleotide in a sequence to the total number of amino acid or nucleotide, while similarity is the degree of resemblance between two sequences. Unlike identity, similarity is not as simple to calculate. Before similarity can be determined, the degree of similarity of the building block of sequences to each other must first be determined. This is done using similarity matrix that are also known as substitution or scoring matrix. Sequence of two homologous proteins can have a similarity of 60% and identity of 40%. They do not show 60% or 40% homology. It should be noted that similarity can be used only for amino acid sequence and not for nucleotide sequence. Before deciding upon the identity or similarity of two nucleotide or amino acid sequences, an alignment must first be calculated. The underlying principle of such an alignment is relatively simple as shown in Figure 1. First, identical sequences. Identical sequences refer to the matched sequences shown as follows. Second, match and mismatch. Match shaded in green refer to the identical sequences. Mismatch shaded in red refer to the different sequences. Third, insertion and deletion. Insertion color in green, deletion color in red. Lastly, conservation mutation as follows. In sequence alignment and nucleotide and amino acid sequences, two sequences are arbitrarily placed next to each other and the alignment judged according to the quality measure. Two sequences are then moved relative to one another, and for each position a score is calculated. This process is repeated until the best alignment is found. Aligning DNA versus protein. The determination of the quality measure is the real challenge for this process, where scoring metric allows the computation of the optimal alignment. A use of identity metric for the construction of alignment nucleotide alignment. For nucleotide sequence, the simplest solution is an identity metric. Here, one assumes that the four nucleotides do not show any similarity to one another, and therefore only identical nucleotides are factored into similarity scoring. They are regarded as identical or different. For the final scoring, only identical nucleotides are added up. B. Use of the Blossom-62 matrix for the construction of an optimal amino acid alignment. Two potential alignments for each are represented. The optimal alignment is shown in grid. For protein sequence, an identity matrix is not sufficient to describe biological and evolutionary processes. Amino acids are not exchanged with the same probability as might be conceived theoretically. For example, an exchange of aspartic acid for glutamic acid is frequently observed. However, a change from aspartic acid to tryptophan is rarely seen. Therefore, most algorithms use substitution matrix to align protein sequences. This amino acid substitution matrix describes a probability that amino acid will be exchanged in the course of evolution. Alignment can be carried out both globally and locally. In global alignment, complete nucleotide or protein sequences are compared to one another over the entire length of the sequence. However, even very similar sequences can have a single deletion or insertion and consequently a different number of amino acid or nucleotide. To represent this alignment appropriately, gap must be inserted into the sequences. Theoretically, all possible sequences can be aligned by introduction of gap. Local alignment. Sometimes interest may focus solely on aligning the most similar strands within two sequences. This approach makes it possible to identify protein domain and motif. For example, ATP binding site, DNA binding domain, and glycosylation site. To compare more than two nucleotide or protein sequence, one could compare all the sequences pairwise and then further examine this alignment. However, it's quicker to perform a multiple alignment and analyze all the alignments. One well-known program is Cluster W, which was subsequently superseded by Cluster Omega. This program utilized the fact that similar sequences are usually homologous. The basic for multiple sequence alignment are the pairwise alignment of all the sequences. This figure shows multiple sequence alignment of 4 related proteins. Amino acid conserved in all 4 sequences are highlighted in green. Those conserved only in 3 or 4 sequences are shaded in red. A frequently used application of pairwise alignment is the search for similar protein or nucleotide sequence in sequence databases. BLAST find region of similarity between biological sequences. The program compare nucleotide or protein sequences to sequence databases and calculate the statistical significance, employ heuristic algorithm. Heuristic method make assessment to obtain almost exact result and utilize sequence and alignment statistics to make searches in larger databases feasible. BLAST is provided as a web service at NCBI, the AMBLEBI, and the DDBJ. BLAST is usually performed first against a non-redundant database, which is a compilation of entries from different databases. In non-redundant database, multiple entries are removed so that every record is available only once. This database exists for both nucleotide and protein sequences. BLAST offers BLAST-N. Nucleotide BLAST is for nucleotide query sequence comparison to nucleotide sequence database. BLAST-P, protein BLAST is for nucleotide query sequence comparison to nucleotide sequence database. BLAST-Add for translated nucleotide query sequence comparison to protein sequence database. T-BLAST-N is for protein query sequence comparison to translated nucleotide database. This is BLAST-N, using it to enter the query sequence and run the. This is the BLAST result. The description tab lists all the sequences that producing the significant alignments. The graphic summary tab summarizes the number and the length of hits with respect to the query sequence. The quality alignment score of the hit is represented by color coding. Alignment tab shows all the significant alignment between query and subject. Actually, there are also a number of species-specific BLAST applications for human, microbial, and other genomes, as well as for the analysis of expressions of immunological data and other special cases. These are all available at NCBI BLAST webpage. So, that's all for me regarding the basic of sequence alignment. Thank you.